Stop, come and take one. Okay, just relax now.
Yep.
Just,
Take one out of 100.
no, it's just going to be one long take. Hi, good to see you again.
Yep.
Let's start with why did you move? Look, let's start with your early life and how you came to be a scientist. Why?
Mm-hmm. Yeah. Right. Well, I grew up in California, kind of Northern California area we refer to as the Bay Area or Silicon Valley. It's where in the 70s, two high school guys made this little box and put an Apple logo on it. That's exactly where I grew up. And then went to San Diego State University, studied chemistry there. Then from there, went to Caltech, continued chemistry for a PhD. It was a little indirect how I got into chemistry. So my dad was a pilot. You'd all remember Pan Am. You're old enough to remember Pan Am. So Dad, he flew the first Pacific routes from San Francisco down to Australia in the late '60s. Anyway, so I always just had this kind of fascination with flight and technical aspects of that. So when I did go to San Diego, my original intention was to do engineering, for aerodynamics, aerospace, that kind of stuff. But of course, first to get into engineering, you had to get the chemistry, the general chemistry out of the way, the calculus, the physics. So that was my first courses, my first year. And I guess, I took well to chemistry. I was enjoying myself. And, my lab TA is like, well, Scott, what are you majoring in? I said, well, I want to do engineering, but dun-dun-dun-dun-dun. And it's like, why don't you do chemistry? It's like, yeah, why not? Yeah, okay, I'll do chemistry. And I guess, I don't know, it's about as much thought went into it. It just felt right, I guess. And my second year, it was just my second year of college, but my lab TA, who was a senior PhD student there, he's like, Scott, you come work with me, like come in the lab, work with him. And, that was like my first taste of real bench research, not the teaching lab experiments, but genuine, research. It's just my second year at university, but that was it. I never looked back. I was hooked. And so, now here I routinely have undergraduates I welcome in the lab and, mentor them for the summer just because, it's important to give back. Because, so that's how I got into chemistry. Again, it wasn't this real planned, deterministic thing. My second year of college, my lab TA, I was doing organic chemistry, and his research lab was just right next door, and he invited me, to kind of took me under his wing for a summer, and I was hooked. I just thought it was just the most amazing thing, and that was only my second year, so I had a couple more years to continue working in that lab with him, and then another mentor, as he left. But I just loved it, and I guess when it did come time to graduate, so at San Diego State, it was actually a good place to get an undergraduate education in chemistry because it was a very small department. We would have 10 or 15 chemistry majors a year. So there was that opportunity for, a lot of mentorship, a lot of time in lab, and really a good place to learn the fundamentals. Now, several of my classmates, came time to graduate, they were keen on just getting jobs like with an undergraduate degree. It often meant, working in like a quality control lab in the local industry, which for me just was not exciting. And so, I was at that point, I think, I really want to stay in science, in research. And my research advisor, the lab I was working in, so he did his PhD at Caltech, back in the 60s, and he said, "Oh, Scott, should, you're dedicated to research. I think you should go to Caltech." It's like, okay. So I wrote to Caltech, and they took me. And so again, what was really valuable in that sense, it was having research experience. That's what Caltech really valued, because PhD, it's basically you're at the bench. You're learning at that level. You're learning from hands-on through your own discovery, and you're learning from other people. The days of learning from books, it's kind of behind you at that level. And so, look, my undergraduate grades were okay. Certainly for some of the chemistry subjects, I struggled. I really had to work to get some, reasonably decent grades. But at Caltech, that wasn't the ultimate priority. They weren't after necessarily straight A people. They wanted people that had research experience and can, work independently at the bench. To succeed, I think you need to be kind of independent at the bench. Of course, there's mentorship, but day in, day out, you're going to the bench. You're kind of working to some extent on your own, making your own discoveries or finding out what doesn't work. But the people that are most successful there, didn't come from these, Ivy League schools or these straight A whizzes. Most of the people that were most successful, they came from, this state school or that state school, this state university. They were able to work independently and work hard. That's probably the most important aspect of research. Research is inherently hard, and so you have to work hard. You don't have to be brilliant. Doesn't hurt, but look, it's a myth that scientists are these just intellectual brilliant minds. It's sure there are some, but that's not, in my view, that's not a requirement to be successful at research. The other question.
That's right. It's like your philosophy in research. Like you said, I loved it.
Yeah.
Why?
Yeah. The intellectual challenge. I mean, it's never a dull moment. Now, for sure, research, so inherent in research is a lot of failure. A lot of our hypotheses, we find they're wrong. A lot of experiments we try, oh, that didn't work. Okay, one step back, two steps back, one step forward. So you do need some kind of like resilience to make it into research, for the long haul. We tell the new students, if you thrive on instant gratification, don't do research. You really need to be in it for the long haul to make real transformative change. So certainly, intellectually, it's amazing. The saying, a lot of scientists will say, my job is my hobby, and for a lot of them, and that's true. And for me, I feel fortunate. I would consider that true as well. And so, it can be very exciting. And when you do come across that moment when you make that discovery, you find something at that point, you're the only one that knows it. No one else knows this. I discovered this. It's addicting. It doesn't happen very often, but usually often enough. It's addicting, and it's enough to, despite the slog, it keeps you coming back for more. You in going about your research, you have some hypothesis. You design the experiment to determine if that hypothesis is right or not right. And the reality is, most of the time, your hypotheses are not right. Okay, because in research we're dealing with the unknown, right? So we're sort of fumbling along in a sophisticated manner, so to speak. But I think the beauty of research is that if you design your experiment properly, such that regardless if your hypothesis is wrong, the outcome of the experiment is still meaningful. And so it's not really a failure. It's, maybe it wasn't what you were thinking. Okay, so yeah, research can sometimes make you feel stupid, but that's part of it, and you get used to it. And so, and after a while, and look, I would say I've taken on some pretty ambitious research projects, for my PhD and so on, and there were times where, months and months of just nothing. It's like, oh, this stinks. But in each case, I eventually figured it out. I overcame, like, if there was a technical issue I had to overcome. And so with that, comes confidence that, I have the confidence to, take on the tough questions, the important questions, no matter how tough they are, and stick with it. And so for new students, that can be challenging. One thing about being at Caltech that I think just rubs off on you, first of all, just the science buzz at Caltech was just amazing. It was addicting.
Is there much sharing of information between them?
Oh yeah. Oh yeah, Caltech was it was a friendly, open place. There's competition in science, but I'm usually not within an institution. I still have very close friends from my Caltech days. That was, what year is it? That was 34 years ago. I got to Caltech in 1990. I was there from 1990 through 1995. The science buzz was, especially coming from, a little San Diego State, kind of mostly undergraduate kind of program. It was a bit overwhelming initially. I think I felt a little out of my depth initially. But just the buzz, and also the faculty there, they just have this fearlessness about them that doesn't matter how challenging or even far-fetched their ideas and research goals are, they go after them. And I think I'd like to think that's rubbed off on me. And that, I think like what I'm doing now with this enzyme telomerase.
Bring us around to that.
Telomerase, yes.
You got trapped into
Yeah.
research.
Yeah.
You're not trapped, you jumped willingly
Yes.
into research.
Yes.
What the attraction was, and what path, how did you end up here?
Here.
And what did you end up studying?
Yes. Okay, so after Caltech, so you do a PhD, and then often you'll do what's called a postdoctoral research. So you move to another lab, usually another area of science, and it's sort of just more advanced, more independent training. And actually, a lot of scientists will say the postdoc years are like the best years in science, because by that time, you know what you're doing. You don't have to do a thesis. You really just get to jump in and research really what you're interested in. It's much more focused. You got more independence. And so I went to a very famous RNA lab in beautiful Boulder, Colorado.
You have to explain what RNA is.
Yes, okay. Can I assume, do we know what DNA is? Can I assume the audience knows what DNA is? So DNA, well, all right. DNA is our genetic material, our genetic code. DNA is composed of 4 building blocks, or what we can refer to as like 4 letters. And the arrangement of these 4 letters determines, who we are. DNA, so all living organisms on life have DNA, have these same 4 building blocks. It's the order of those 4 building blocks that determine if you're a human or the cicadas that are making a buzz outside. Okay?
Yeah.
Now RNA is structurally at the chemical level, RNA is almost identical to DNA. It's difference of just 1 atom. So RNA has the same genetic information as DNA. But what RNA can do, so RNA can also promote chemical reactions in the cell. And so it has this dual function. It's sort of the bridge between the genetic information and actually performing cellular functions. Now, when this was discovered in the mid-'80s by Professor Tom Chuck at the University of Colorado, it really just changed how we thought about biology. I mean, it rewrote textbooks, and he was promptly awarded the Nobel Prize for that discovery. So I went to his lab in 1995 to learn some RNA chemistry, and yeah, it was an amazing lab.
Must have been exciting.
It was. I think what made the lab so special was that he had people from all different backgrounds. So he had me, sort of the full-on chemistry geek. He had molecular biologists, cell biologists, geneticists, each looking at different aspects of RNA biology or chemistry, and so being exposed to that diverse pool of talent. And I still think back sometimes that maybe I didn't take advantage of that enough at the time, because I knew chemistry. I never really at that point knew much biology, and so I mean, looking back, it's easy to say in hindsight, but I probably could have maybe picked more brains than I already did.
It's always easy in hindsight.
Yeah, it is. And you need to be careful not to torture yourself in that way as well. But yeah, it was scientifically, it was amazing. At a personal level, I still have many very close friends from those days. And when I visit Boulder routinely. And so now relating to
So
Sydney,
right back to you,
Australia.
So RNA. So
RNA.
that's when you met RNA,
Yes.
and RNA met you.
Yep. And what else I met in the lab? I'd been there a couple years, and after a couple years, Tom, he hired this cute postdoc who just completed her PhD here at Children's Medical Research Institute in cancer cell biology in Roger's lab, Tracy Bryan. She came to the lab, and the rest is history, I guess. What often happens is the Aussie girls come when they're cute accent and move in for the kill, and and we've been here 24 years. So
So was it through Tracy that you ended up doing
That's-
cancer?
yes, and so
You might want to say it was through Tracy, because my voice is not going to appear. on
yeah,
this.
right. And so so Tracy and I we hit it off pretty well. Ironically, we didn't really have much in common at the time. So her PhD was in cancer cell biology, and I was doing more hardcore RNA chemistry. But Boulder's the kind of place where we had the same outdoor interest, and that being hiking in the beautiful mountains there. And so now Tracy's PhD here, she hugely successful, very important PhD, and when she left CMRI, she had a standing offer to come back as her independent lab head. So in 2001, we moved here to Sydney, and she started her independent program. And initially, I was at the University of Sydney for a little bit, but then thought, why don't I come here and learn biology? And with the connection with Roger, um, and his interest also in telomeres and telomerase and cancer biology. So Roger kindly offered me I got a bench and why don't you come in my lab? And so it just kind of seemed right, just have a natural fit. And so that worked out well. Um, and so that's how I got into telomerase. It wasn't until I came here. Now, in the RNA lab in Colorado, when Tracy got to the lab, actually telomerase was becoming a important research area at that time.
Well, we can't dodge this much longer, you know. What a telomerase.
Oh, telomerase and telomeres. Yes.
Okay, they sound the same.
Yes, I will
Sound
try
the
to.
same when pronounced
I will
telomere.
try to enunciate better and hope my accent doesn't.
No, it's not that. It's to my ear. I'm not sure the difference.
Mm-hmm.
But they sound basically the same. They
Yeah.
come from the same source, but with
Yep.
different.
Yep.
I don't know.
Yeah.
And
Okay.
I'm an idiot.
Telomeres. Telomeres is sort of a fancy name for the ends of our DNA chromosomes. So if you imagine our chromosomes like essentially a piece of string. So humans, we have 46 chromosomes. So piece of string, each chromosome has two ends. Now the ends are kind of special, and those are what we call telomeres. Okay. Now an important feature of telomeres actually shorten each time a cell divides. And so as we age, our telomeres are shortening. And that's normal. That's sort of kind of nature's timepiece. It's nature's counting mechanism to, limit cellular growth. Right? Look at us. We're symmetrical, we're defined proportions. So built into our genes are these, inherent limits of how many times our cells divide. And that's regulated, at least partly, by the telomeres, which are shortening, okay, as we age. Now, the connection with cancer, I guess the defining characteristic of cancer, so cancer is kind of a collective term. There's cancers of all of our different tissues and organs, and so, I think there's something like almost 200 different diseases that are collectively referred to as cancer. But I think the unifying, defining property is that cancer cells, they've acquired enough genetic mutations that they can bypass these built-in limits of cell proliferation, and so cancer cells divide without limit. In the laboratory, we actually call these cells immortal. They truly are immortal.
So the normal ones divide and shorten.
So normal cells divide kind of a finite number of times, and their telomeres shorten. Now, when a cancer cell divides, its telomeres also shorten a little bit. But this is where telomerase comes in, okay? So telomere, that's the end that's the DNA. Telomerase is what we call an enzyme, essentially a molecular machine. Our cells, we have thousands of different enzymes. Each one performs a particular role that keeps our cells going, that carries out the chemistry of life. Now, what telomerase does, telomerase lengthens telomeres. So in cancer cells, most cancer cells have turned on telomerase. If we go back to our normal cells, most of our normal cells, there's no telomerase. It's undetectable. Our telomeres shorten. Okay, that's normal. Cancer cells, with their profile of genetic mutations, they can break the rules, and one thing they do, they turn on telomerase. So when a cancer cell divides, sure, its telomeres are going to shorten, but telomerase is right there to re-lengthen it. So over time, we do this in the lab. If we take normal cells, over a couple months, we can measure their telomeres in the lab, and we see this progressive shortening. Telomeres of the cancer cells are just They don't change. And so in cancer research, what's been done for many decades now is that um, a human tumor, under the careful right conditions, sometimes these cells from that tumor can be propagated in the laboratory indefinitely. Okay, we call that it's a cancer-derived cell line. So that's sort of the most fundamental sort of research tool we use at the cellular level in cancer. We have cell lines derived from breast cancer, from lung cancer, from brain cancer, just about every kind of cancer. We now have these cell lines that we can grow in the laboratory. And so that's our model for either understanding cancer cell biology, testing hypotheses, testing new treatments.
So it's almost like, what do they call it with sourdough bread? You have the yeast, like
The starter.
yeah.
Yes, I make sourdough bread.
So is it like that? Is that not dissimilar?
Well, you're right, it is. And that, we have these cells, they grow indefinitely. So we call it a flask. We have cells, they grow. After a few days, they essentially filled up the flask. So then what we do, we take some, we put it into a new flask, and they keep going. And there are cell lines that have been going for 4 or 5 decades in labs all over the world. And so
what do you do with this now?
So if we go back to telomerase, so telomerase, it's a promising target to develop future cancer therapies, because about 90% of all cancers, representing pretty much every organ and tissue type, about 90% of all cancers have turned on telomerase to support their unlimited proliferation. So then the idea is, in its simplified form, that if we can develop a therapy that shuts down telomerase, what we would call a telomerase inhibitor, if we can shut down telomerase, then presumably the telomeres of the cancer cells will then shorten, and that will limit or stop or at least limit the growth of the cancer. So that's the basic hypothesis with respect to targeting telomerase as a cancer therapy. Now, so it's 2024, that really developed, the association between telomerase and cancer was established in 1994. So it's been 30 years. And I guess that kind of shows science can be slow. And
you can say that, but over 3,000 years.
Well, sure.
Yeah.
Yes, in that sense, right, it's been a blink of an eye, I guess you could say.
What would you say,
but-
because from my observation and talking to people, the last 30 years and the leap forward in cancer research has been phenomenal, considering
I
what's happened.
agree, but that's it. That's the time frame you need to think about, almost like generational, or certainly on the decade time frame. Even just year by year, sometimes it can be, as a researcher, hard to really see major progress. But for, I think, real transformative change, you're looking on the decade timescale. And I think for developing new therapies, so for example, medications that everyone takes now, I take them for granted, that was research that was done probably in the at least 70s and 80s. Typically in drug development, you're looking at 2 decades maybe of laboratory research, followed by another decade, as progressing through clinical trials. You're looking at least 2, typically 3 decades from the beginning basic research to, having a therapy.
From inception to a possible result.
Exactly. And yeah, it's a tough road. It's fraught with failure.
But once again, is it failure?
Well, I would say, yeah, at the basic research level, that discovery-based research, no, failure is not the word. There are, many instances. So if we look at the clinical path, as we, move from what we call fundamental research, gathering knowledge to, applying that knowledge to the clinic. So there's clinical trials, there's phase 1, phase 2, and phase 3. So phase 1, it's usually 10 or 20 patients, and really the only objective is, can we put this thing into a human safely? The question isn't how well does it work, it's is it safe? Phase 2, you finally get to, can we treat disease with this therapy? And that'll be a larger trial, maybe 50 to 100. And then phase 3, you'd be maybe 1,000 or a few thousand patients. And so also with that, phase 1, you're looking at these days, I don't know, 20 million. Phase 2, 50 to 100 million. A phase 3 trial, a few hundred million. And so there, maybe the word failure might be appropriate. There are, most don't make it through phase 1 and 2. And that, yeah, you're talking big money and, a lot of huge investment.
Is that because the research was deemed to be advancing, or cut your losses and run?
Probably both. I mean, there's the science, and then there's the whole business case as well. There are instances where, despite doing great, the laboratory discovery-based research, sometimes putting things into a human, there are things you don't foresee. Unexpected toxicity. And it never makes it past phase 1. That happens a lot. And, there's this element of things you just don't foresee. For me, so I've been studying telomerase for 3 years now. And I'm mostly still at the level of basic discovery-based research. But I feel I'm kind of now making or about to make that transition to more applied research. So I mentioned the term telomerase inhibitor, right? So a molecule that can shut off telomerase as potential therapy for used in cancer. So I have found one. It took it took at least 15 years, and followed by another 5 years just to kind of characterize it and really make sure it works. It works great in the lab. Is it ready to be put into a living, breathing organism? Starting with a laboratory mouse? No, not yet. Okay, not yet. It's
What's required to get to that stage?
So there's a few characteristics in terms of developing a therapeutic. We use the term potency. So the more potent, the lower the dose required. And potency refers to, if we let's use the lock and key analogy of a telomerase inhibitor, or more generally, an enzyme inhibitor. So telomerase is the lock, and your key you want to fit in snugly and uniquely to the lock. Okay? Uniquely, so it's going to target telomerase, but it's not going to mess with anything else in the cell. Okay? In chemistry, we call that specificity. And specificity at that molecular level will translate to a safer therapy at the clinical level. Okay, so many side effects of therapies occur because the drug is interacting with other molecules that are it's not intended to. That happens. Okay, and so we'd like to understand and optimize this potency and this snugness of fit at the molecular level. And that's going to give us, or should give us, hopefully, a safer therapy at the clinical level. Okay, so we'd like to improve that. And then there's another concept referred to as medicinal chemistry, where a lot of drugs that we take, at the chemical level, there are certain properties that many drugs have that are desirable. Things that affect, it's how well it, how well it's, how long it lasts in the body. Does it break down quickly? Other properties like that. Can it simply be, compressed into a little pill form, taken orally?
How many basic drugs are there? I mean,
Oh, how many drugs are there?
Families, of course, they're
Oh.
families, aren't they? So you've got one and there's offshoots.
Oh,
I'm wrong.
Yeah,
I don't know.
I mean, there's countless, drugs that would be defined as a small organic molecule that, they started in the laboratory. They started probably with biochemistry and maybe worked their way into cells in the laboratory and then into a laboratory mouse. And along the way, they, what that first molecule was probably modified through chemical synthesis to chemical synthesis. So what chemists are good at, we can make cool things and get them pure usually too. So that's what chemists do. We make things. Chemical synthesis is the discipline of assembling molecules. Okay, so many of the big drug companies will have hundreds of chemists that synthesize their drugs and drug candidates.
And that's synthetic drugs. So if you go back to opioids or something like that, so that's sort of natural start. So
that's right.
just talk me through those two. Do you replicate that in the laboratory, or?
Can you
repeat?
repeat?
A synthetic opioid.
Yeah. So.
This might not
Right.
be a documentary, and I'm not trying to develop that. I'm not doing a.
Look, I mean, so you're right. So some drugs are actually many drugs are derived from what we call natural products. So Taxol used in chemotherapy is derived from the bark of a tree in the Pacific Northwest. It's a finite resource, so then chemists try to produce that in the laboratory through chemical synthesis, starting with small molecular building blocks and then assembling them to the more complex molecule that is Taxol. And that's done for many drugs.
That's a good example.
Mm-hmm. Yep.
Excuse me. So how you going?
Yeah. Look at if I look back over the last several years, it making progress. Again, I'd like to think I'm kind of at that inflection point where maybe getting into the applied, and that we have a candidate, we're at the point where we want to tweak it just to get it just right, and move on to more complex experiments. So right now I'm doing mostly the biochemistry. So in a test tube.
Sorry, yes, I was hoping you didn't notice it.
Yeah. Right. So this telomerase inhibitor I'm working with look, it has some enticing properties. Currently, we're doing biochemistry with it primarily. So, in a test tube. We're about ready soon to move into, these immortal cells we work with in the lab, and there's lots of questions and hypotheses to explore there, and then from there you'd move into a laboratory mouse. Experiments sort of get more and more expensive as you go on, so you don't want to be too hasty. But for example, so of course I collaborate with Tracy. Tracy's also studying telomerase, more from the cell biology perspective. And so one hypothesis we want to explore, and this is based on some of the discoveries from her lab, in that we hypothesize that actually combining a telomerase inhibitor with standard chemotherapy that's used next door will actually allow lower doses of the chemotherapy to be used. Okay, they'll kind of augment each other, or more bang for your buck. And lower doses of anything invariably is improved safety, fewer side effects, and certainly for chemotherapy, that's pretty important. So that's, for example, that's one hypothesis we'd like to test with our telomerase inhibitor. So
I think you explained that really well.
okay,
I understood.
yeah,
Do you understand?
yeah, all right, yeah. So it's
If you've got any
inspiration.
any research in the past where you went, holy crap, that's fantastic, or that
Yeah.
came out of left field, or even from, I don't know.
Yeah.
I sort of got turned on with cowpox and smallpox. I thought that was a hell of
Uh-huh.
a leap.
Yeah.
I was just wondering, that was just me. I was just wondering if you had any
Kind of like
favourite,
moments.
personal moments, but also favourite moments in the history of research.
Oh, in the history of science.
Yeah. I know, I've hit you out of the blue with that one, but
Yeah.
whether it would resonate or not. So it's like,
Well, I'll tell you about one of my kind of coolest things I developed, or I discovered. So my first task when I got here, so in 2002-3, look, the association of telomerase and cancer was pretty well established. But as a chemist, to me there are questions that still need to be addressed, like what is telomerase? At the molecular level, what are the parts that put it together, and how is it put together? So my first task, something that had never been done, was to obtain a pure sample of telomerase that you could then analyze properly. And so my first task was to essentially extract the telomerase from immortal cells we can grow in culture, and it required a few different steps along the way, and I was kind of stuck at one last step. I had one more step to go, but I was essentially stuck in that, and literally the enzyme was stuck. It was stuck on the reagent I was using during the purification, and so this went on for a few weeks. Like, darn, I've come all this way. And a couple weeks later, lying awake at 2 a.m., of course, it's so simple. And I guess, well, that's where hindsight is, right? The simplicity was actually just, or I think that it was described by others, not me, it was described as elegant. And its elegance was in its simplicity. And that, without delving in the details, it was actually It was incredibly satisfying. And to this day, it's probably one of the coolest things I've ever done with this enzyme.
Come on, delve into the detail of it, even if you
All
don't
right,
use it
I'm
and it's
gonna
not. Well,
I'm gonna
we'll
try it.
record it. Go on.
All right. So in one method in enzyme purification, it's what we call affinity reagents. Imagine little beads, little plastic beads that and beads are handy because you can collect them, in a cup or a filter. And we can modify these beads with different molecules. So what I did, I modified the beads with a piece of telomere. Okay, so using chemical synthesis, I took a little plastic bead and modified it with a piece of telomere. Okay, and then I combined this with the telomerase from the immortal cells. All right, and the telomerase binds the telomere, and then I collect the beads, and that's how we can extract the telomerase away from everything else. And these things are really effective. You can get 1,000-fold enrichment in a single step. And okay, so now the thing is, telomerase bound this piece of telomere really, really well, super tight. It just wouldn't let go. Now that was great because I recovered almost all of the telomerase. Okay, but then I was stuck. I couldn't get it off. Like, okay, so I'm really stuck here. I got my telomerase on a bead, and I can't do anything with him. Step one complete. And that's the thing, purification, it's always a balance of opposing forces, right? The capture worked super well, super clean. Okay, and then so this went on for a few weeks. Oh, how can I get it off? How can I get it off? Well, there's this cool property of telomerase that if the enzyme undergoes a round of, what in chemistry we call it catalysis. So essentially making it add a piece of telomere. So the enzyme stuck to a piece of telomere. What I did is make it extend the telomere even further. And in doing so, it actually falls off. And so what that means is that I was able to collect the telomerase, only the telomerase that was active and fully assembled, the perfect enzyme. And it's actually very rare in enzyme purification to make that distinction, to collect only active, fully assembled molecules. And so that came, it was like 2 a.m. one night, lying awake. It's like, oh, I can't believe this. And in hindsight, it was really simple, and it just really illustrated the elegance of the enzyme, of how the enzyme works.
But you were lateral enough to say, if I did this, it'll fall off.
Yes, and it did. It worked really well. And I guess that was, I still kind of...
That's a Yahoo moment.
It was, so that was, what some scientists will say, just that sense of wonder, of really understanding nature and seeing it. When we don't understand something, we think, oh, this is complicated. But then once we understand it, like, yeah, of course, it's simple. Nature's always simple, and everything makes sense, and it's elegant. You're just making that transition.
Everything's simple when you know.
Exactly. And so that was pretty cool. And I'm kind of, at least in the telomerase field, so reasonably it's a little bit well known for that one. That was pretty cool. So
any other piece of research historically that you found fascinating? Not so much because it was just part of the study you had to do.
Gosh.
That's that one that's fine.
Yeah. Sorry. Yeah. Nothing.
It's not penicillin.
Yeah. Nothing comes to mind. Penicillin. It was an Australian-born chemist that discovered penicillin, or identified the molecular structure that we know as penicillin. So Howard Florey from Adelaide, he was educated in Adelaide as a chemist, or as you say here, like a pharmacist. He was working at Oxford, and identified the molecule that is penicillin, and then he developed the means to mass produce it on the society level. This was, I think, late 30s, so just before World War II.
I believe there was a huge amount of money put towards it.
Well,
by the
yeah,
US government.
yes. As World War II got going, I mean, yeah, he single-handedly saved probably millions of lives just in those first few years through antibiotics, through penicillin. And so,
Fleming discovered it in the petri dish. and
correct, correct. Yeah,
What would you say? Honed it.
yeah, honed it. So, and telomerase. I'll tell you about how telomerase was found. Like many things, telomerase also started as a scientific curiosity. In the, I guess, from the 60s, 70s through the 80s, what a lot of cell biologists were working with as a model organism, not human cells, because human cells, they grow slow, they're really expensive to culture, it's not easy. They're using single-celled protozoa. So maybe in high school biology, did you put a drop of pond water under the microscope and look under the microscope and you might see things kind of swimming around? And those are single-celled protozoa. At the macroscopic level, they don't look much like a human, but at the molecular level, you'd be surprised. A lot of things are pretty similar. And again, if thinking about DNA, they have the same 4 building blocks in their DNA as we do. It's just the order of those building blocks is different. Now, if you think about a single-celled organism, one divides into 2, they swim away, those divide, and as single-celled organisms must divide without limit. So at a conceptual level, you can think of them as immortal, kind of like the cancer cells we talked about earlier. And when a single-celled protozoa divides, again, its telomeres also shorten. That telomere shortening, it's an inherent property of the process of cell division. So, and I think a key question around the 70s was, well, how do these little things maintain their telomeres? Why don't their telomeres just shorten to nothing? And so as an Australian scientist, Elizabeth Blackburn from Hobart, this was during her research career in the States. She was at UC Berkeley. She thought, to her credit, well, maybe there's a special enzyme that's what it does. It lengthens telomeres. And so she told her PhD student, Carol Greider, well, that's your thesis, go find it. And they did. They found it, and their landmark paper in 1985, it's considered the birth of the telomerase field. It's the first scientific report showing that this kind of enzyme exists. And so the connection with cancer, human cancer, so that landmark paper was 1994. It's almost a decade later. And that is really typical. I mean, telomerase is one of who knows how many different stories or different therapies or technologies that we might take for granted now, but decades before they started as a scientific curiosity. And I think that illustrates the importance of just fundamental curiosity-driven research. What some scientists might get frustrated these days, sometimes there's this attitude maybe among our elected officials that, well, we need a return, we need a guaranteed return on our investment in science. And it's like, well, it doesn't work that way. I mean, yes, it's well established that a dollar spent now decade later, it will, you know, recoup and much more the cost. But you know, you can't put a time frame on it, and you don't want to. You can't necessarily direct science. You can't. You know, it doesn't work well. It's like, okay, go invent this. It doesn't really work well that way. And I think one challenge for research scientists with respect to research funding, there's this kind of inherent disconnect between the time frame of, you know, real impactful progress in research, you know, a decade or decades plural, you know, and the time frame of a term of public office, which is a few years. And often research, you know, public officials, they dedicate money to research. Those benefits, you know, will probably be well when they're gone. And so it does take, you know, some courage and vision to invest in research now, realizing that, you know, well, those benefits I may not be around when that real impact happens. So I guess, look, I don't want to. No one likes a whinger, but I will say that, you know, research funding here, it's a bit lean. It's a struggle. I mean, you know, if you ask me how things are going, it's like a little more funding would be good. You know, but I think at a higher level, there's another reason why research funding is important. So we're next door to a children's hospital, and you know, Tracy and I were very local. I walk 35-minute that way. That's home. Our walk to and from work, we go through the kids' hospital. Now, walking through a children's hospital is the ultimate reality check. You think you're having a bad day, you walk through the kids' hospital, and we walk just down the hall from the oncology ward. It's like, yeah, okay, I got my health. It's okay. You know, maybe my problems aren't so bad. And so for, you know, while you might be fair to, you know, say scientists are being too self-serving when they say, oh, we could use more funding. But at a higher level, so for patients and their families, research gives them hope. Okay, and that's what you're giving when you support scientific research at any level, whether it's philanthropic or, you know, an elected official, you know, putting hundreds of millions of dollars towards research. You're giving the patients and their families hope that there may be a better treatment, a better future. And that's important. And, you know, look at the day-to-day at the lab bench. Yeah, what drives me, look, I'm a geek, I'm a science geek, I love it. But, I mean, look, at the data level, what mostly drives me is, look, I'm a science geek, I love it, okay? I'm not ashamed to admit that. But, you know, at a higher level, certainly, again, walking to and through the kids' hospital twice a day, I'm reminded that, yeah, I like to think I'm contributing to the common good, and it would be amazing if my research actually made it into the clinic. I mean, look, I realize it's a long road, it's a bit of a long shot, but I think nonetheless, it is satisfying that as a scientist, I'm contributing to the common good. I'm giving a reason that, you know, the patients next door, you know, might have hope that we can develop better things, better treatments. And there will be better treatments. It takes time. Yeah, you know, look, and it's not just medical research, but more the technology, devices we use every day. You know, the space program in the United States, starting in the 50s, certainly the 60s, Computers used to be the size of this room. They realized, well, okay, well, we're not getting that up in a rocket. Can we improve on them? And just countless technologies, satellite technology, the technology we use in our phones, in our kitchen, every day at work. A lot of these technologies, they came about through the space program, which was research. That was high risk, high reward research. And so, yeah, look, it's really hard to look at any area of, discovery-based research and, okay, well, is that gonna give me anything in 10 years? You don't know, but you just have to have that faith that, in the grand scheme of things, research, it's going to improve our quality of life. It's going to give us new treatments, and so on. One way to look at research and the outcomes, one description we'd like to use is that of a pyramid. So at the base, the widest part is discovery-based research, curiosity-driven research. No application in mind, just wanting to understand how nature works. Okay? As you go up, you might go to applied, and at the very tip, you'll get to success, new technologies, new treatments, new therapies. And the reason I like this analogy is that you need a very broad base of fundamental research to give you that pipeline of real successful outcomes that change the way we live. Because a lot of basic research does not lead to tangible discoveries that change how we live and so on. And that's why I think the pyramid is a good analogy or a good way to display that. You do need a broad base, and look, only a small fraction of that is going to change the way we live. And at this level, when you're doing the basic research, you cannot predict that. No one can. And communicating that to non-scientists is a big challenge, but very important. So very few scientists, particularly at purely academic scientists like at universities, many scientists are not there to, okay, I'm going to discover this cure. They're there to understand nature. Okay, so mutations are in our genes. So if you imagine a chromosome, the very ends are the telomeres, and in between are many genes and the genes are the instructions for all of the different enzymes that carry out the chemistry of life. And so that's where the mutations are in the genes. So that's the idea to shut off the telomerase, and then the telomeres will shorten in the cancer cells. Okay, then the T cells. So that's a kind of a different realm. That's immunology, immunotherapy, of which I know little.
Oh, right. No, but I was
But-
interested because you were talking about creating telomerase inhibitors.
A telomerase inhibitor. So
Yeah, and that would help stop these cells from mutating or becoming cancerous and stuff.
that would, so the cells are, so the way it would be used, the cells are already cancerous and they have telomerase. Oh, plain. Yep. Okay. To explain how a telomerase inhibitor would be used, it wouldn't be used to prevent cancer. It would be a cancer therapy after the cancer has developed. The cancer cells, they will have telomerase that is allowing them to prevent the telomere shortening, And so administering the telomerase inhibitor would shut off the telomerase, and then the telomeres of the cancer cells would then shorten. So that's how, in its simplest form, a telomerase inhibitor would be used. Exactly, that would kill or at least limit the growth of the cancer cells. So then some of these other hypotheses, for example, that we're looking at, combining the telomerase inhibitor with chemotherapy, that might be a even more effective approach where we're preventing telomerase from lengthening the telomeres, so the telomeres are shortening, but then there's also chemotherapy, which is damaging the DNA, and so those two things going on at the same time may actually be more effective than either one alone. And that concept we call synergy. Look, more bang for your buck. And that if you may have two drugs, you combine them, and they work far better than either one alone. Okay, and that gets down to understanding, again, at the molecular level, what's going on in the cell. How is the DNA being damaged, and so on. So this is where fundamental biochemistry and cell biology come in. Okay, ultimately, things are happening at the molecular level. So chemotherapy, it's damaging the DNA in one way or the other, depending on which drug. We want to understand exactly what molecules are doing what. That's the level of understanding we aim for in cancer research. So the current hypothesis is that yes, extended use of a telomerase inhibitor, the telomeres of the cancer cells are going to shorten, and then eventually those cancer cells will die. That's sort of the ideal, perhaps somewhat simplified hypothesis. Then the key hypothesis with respect to a telomerase inhibitor and cancer. A telomerase inhibitor, it will shut down telomerase, and so the telomeres of the cancer cells will progressively shorten, and eventually those cancer cells will die. That's really the key hypothesis that has been around for 3 decades now.